Okay, so welcome. So today I'm going to tell you about birdsong and the brain. I'm going to tell you about a lot of work that my group has been doing along with a lot of our collaborators over the last few years to try to understand general principles about how the brain controls the body. So, for any skilled behavior, from speaking to dancing to anything, the brain is facing this extremely hard mathematical problem that it has to solve. And we've been using lots of different approaches, including primarily until very recently working on songbirds to try to discover some of these general principles. Okay, so what is the problem that we want to solve here that I alluded to a minute ago? Which is that, for any of the complex behaviors, that we as humans excel at, the brain has to solve this extremely difficult problem, and we as neuroscientists investigating the brain have to solve sort of similarly different problems as we investigate the brain. So this is schematized at left here. So you have millions upon millions upon millions of neurons controlling any particular behavior you might be doing. That behavior is actually affected by hundreds of muscles that are working together in these really complex ways. And then because of the physical biomechanics, how the muscles actually pull on your body, there's this very complicated relationship between muscle activity and behavior. And then something sort of, I think, even more important and magical seeming happens, which is that the sensory signals that arise from behavior are used to rewire your brain during learning. So let's take playing a musical instrument as a great example of this. So as you learn how to play your instrument, your brain is constantly using sensory feedback, primarily what you're hearing, from whatever instrument that you're playing, to actually rearrange the brain, the activity in your brain, and over hundreds of thousands of iterations, you can achieve people can achieve these really expert levels of motor performance. So a lot of them, our lab's work does is, we try to answer these very, very general questions that I'm showing at the right here. And these questions are very linked, as I'm going to talk about in a moment, which is that first we want to understand, how the brain controls these incredibly complicated behaviors. How do millions of neurons, control hundreds of muscles to do, something as elaborate as dancing, say. And then the linked question that we also study is we want to understand how the brain uses sensory information, or what I'm going to call sensory feedback, to correct motor errors, which is another way of saying to improve performance over time. And we combine lots and lots of different scientific approaches to do that. So this is what the cartoon is showing. And as mentioned in the very nice introduction, we combine a lot of stuff. We combine a lot of biological and behavioral approaches. The first experiments I'll talk to you about are just doing very close examinations of behavior. We also use the tools of neuroscience to understand the structure and function of different aspects or different parts of the nervous system. And then we use a lot of mathematical tools, including developing new mathematical tools to try to understand sort of how this all fits together, and how is it that the statistical patterns, the math that the neurons seem to be doing, how does that actually succeed in controlling the particular behaviors that we care about? Okay, so to give you a flavor, for all this, I'm going to walk through a few different kinds of projects in my lab. And I'm going to start with a project that, as I said, is purely behavioral. So we're trying to learn about the brain, or I hope we are learning about the brain, but we're doing this without observing brain activity directly, except through behavior. So I'll talk to you now, and this is actually sort of the branch of neuroscience that I got my PhD in, which we call psychophysics, which is ways of investigating the mathematics of brain function just through looking at behavior. Now, the particular behavior that we look at is the vocal control of birdsong. As I mentioned, this is what I've been working on, birdsong since my postdoctoral fellowship, and until recently, my lab was 100% focused on birdsong. And I think that's with good reason. Songbirds are a really wonderful model system to use to look at the brain mechanisms underlying the control and also the modification, the learning of a really complex behavior. So I'm showing you some examples here of song from a Bengalese finch. This is most of the bird data I'll talk about today is from this species. So let me explain what you're looking at here. This is called a spectrogram. And I'll take a moment to go through this, since I'll be showing a lot of this kind of plot today. So time is on, this is a graph, time is on the horizontal axis. On the vertical axis are the different sound frequencies of sound that can be produced. So high-pitched sounds are high up on the y-axis, lower-pitched sounds are lower down. And the color map there, the pattern of colors that you're seeing, that shows the energy or the amount of sound at each acoustic frequency as a function of time. So if you look at the song of an adult Bengalese finch on the top there, you can see that the bird's song is divided into these discrete vocalizations. I'm going to play you the recording of that in just a moment. And we refer to each one of these vocalizations as a song syllable, and this is very intentionally analogous to syllables in human speech. And in fact, if you look at this, you can see that the same syllable, a lot of the time is being repeated throughout the song. Okay, so I'll get to the recording in a minute, but first I'm going to play the song of an adult. Then I'm going to play the song of one of his sons. So in the particular species that we study, only male birds sing. In our species, they sing both for courtship behavior and also for to establish territoriality. But interestingly, young songbirds are not born knowing how to sing their adult songs. Rather, they are born with this innate learning ability that allows them to copy the vocalizations of whichever members of its own species it has social interactions with, which should, and I assume does, remind you of quite a bit of human speech, which again, no infant is born, knowing how to speak an adult language, but almost every infant has the capacity to mimic or to learn by imitation the language that it hears, its parents or whatever, its family or social group using. So it's just like this in songbirds. So at 51 days old, young songbirds have just begun to vocalize. And as you can see from the spectrogram, and again, as I'll play for you through sound, I hope, in a minute, when he first begins to vocalize, this young bird has a song that sort of looks and sounds nothing like the song of a normal adult. But then on the bottom, this is the bottom row, and the third clip of sound I'm gonna play for you. This is the same young bird after 40 more days of practice. And what's happened in these 40 days is that, first of all, the young bird has memorized the song of the tutor, and then has spent 40 days practicing, listening to all of the mistakes that he's making, and changing his song so that it's a more precise match to the song of the tutor. So now, and this is the part that I have not tested, so I hope you can hear this nicely. Please tell me if you can. I'm going to play these 3 sound clips. First, the father, we also refer to that as the tutor, then the young bird right when it starts to vocalize, then that same young bird after 40 more days of practice when he's learned how to produce a pretty good copy of the tutor song. Then I'm going to go back and play the tutor song one more time. So let's hope this works. Okay, I hope you just heard the song of the tutor. Now listen carefully, the juvenile song is very quiet. All right, now we're going back to that same young bird after 40 days of practice. Then I'm going to go back and replay the tutor song so you can compare. Okay, so I see some people smiling. I hope that means the demo worked. So what I hope you can all see there is that there's this incredible process of vocal imitation, an illustration of this incredible process of vocal imitation. And what I'm showing on the left is sort of the field's current understanding of how this works. Okay, so that big box called the motor system, what I mean there is all of the parts of a songbird's brain and its muscles and its vocal organ that are creating the song. So the idea is that the juvenile bird, the 51-day-old bird, the motor system starts to produce some song, and then the bird is monitoring, listening to the auditory feedback, and then this is that big minus sign there, comparing that auditory feedback with some memorized auditory target or template of what the song is supposed to sound like. And the idea is that that is a copy of the song of the tutor. Then, and this is the important part, the idea is that some sort of subtraction happens, where the bird's brain computes the difference between the feedback, which is what he actually sang, and the target, which is what he wanted to sing, to produce an error signal. And the idea is that that error signal is then somehow used to rewire the motor system to improve the bird's song. Okay, so one of the other things, so I just told you about this incredible process of song learning that young birds do. Now I'll tell you something even more, well, equally interesting, let's say, which is that after this incredible period of change, of plasticity, when the bird is totally remapping its singing patterns, song, if in the under normal circumstances, is then completely stable for the rest of the bird's life. So the juvenile here has totally changed his song in 40 days, but I could come back and record from that same bird years later, and it would have the same or almost exactly the same song that he had at 90 days old. So how do we go about thinking about this? What is the relationship between the process of learning during development, and learning throughout the rest of life? So and this was actually one of the first sets of experiments that I did when I started working on songbirds, which is that we made a hypothesis, which is that basically it was all the same, which is that the same process of comparing auditory feedback to an auditory target that drives or that motivates the initial acquisition of song in a very young bird was also the thing that would allow birds to have this incredible stability of birdsong throughout life. So to figure out a way to test that hypothesis, we had to figure out a way to trick adult birds into thinking that they were making a mistake, to ask if you do that, whether birds will actually change their songs. And this is what I spent the first 2 years of my postdoc doing. I'm not embarrassed to say, I'm the world's leading expert in putting headphones on songbirds. So I spent a couple years figuring out the ergonomics of this. My parents are still proud. Okay, but here's why we went to the trouble, which is that it's not just that we put cute headphones on songbirds, it's that we put cute headphones on songbirds and then use them to manipulate birds' auditory feedback. We used the system to trick birds into thinking that they were singing incorrectly. So the way the system works is that there's a microphone in the bird's cage, bird sings, and then we have fancy, very expensive online sound processing hardware that can manipulate the bird's song in real time, and then it's played with a very, very short delay back through the headphones. So the idea is that we're actually replacing the bird's actual auditory feedback with some manipulated version. And this is the crucial thing here, is that we can control the error that the bird is receiving. And in fact, the first experiments we did were very simple, is we just shifted the pitch of how the birds thought that they were singing, or of the sound of the bird's song. So in effect, what we did is we used the headphones to make the birds think that they were singing either one semitone too high or one semitone too low. So musicians, you'll know what a semitone is. Non-musicians, it's the pitch interval between two adjacent keys on a piano keyboard. The mathematically oriented, a semitone is about a 6% change in the absolute frequency of the sound. So we introduced this one semitone error into a bird's auditory feedback to see if an adult songbird, who has this beautifully crystallized song that he's already sung 1 million times before, to see whether they'll continue to use this error correction. So the way we did this is the spectrogram on the left here. This shows a song from one bird, and we used math to go and repeatedly measure each time, for example, syllable A was sung. We would measure the pitch of that syllable. And then over the course of time, we could measure how the pitch of these different syllables changed in response to this manipulation we were doing of how the bird hears himself sing. Okay, so on to the data on the right. So this was an experiment where we let the bird get used to the headphones with 0 pitch shift. Then all of a sudden, we increased the pitch of how the bird hears himself by 1 semitone and measured the resulting changes in behavior. So here, the colored lines at the bottom there, that shows the change in pitch of those 4 individual syllables I'm showing on the left. And let's just look over the first 14 days here. So you'll see that in response to increasing the pitch of how a bird hears himself, this bird is systematically lowering the pitch of all of the different syllables in his repertoire. And one thing I want to point out here is you see the dashed line up there that says heard. I just want to clarify something. Is the way that these pitch shifts work is that they are always relative to the bird's current vocal production. So what that means is that when we first turn the pitch shift on, the bird experiences a 1 semitone error. His song sounds 1 semitone off to him. But then by lowering the pitch of his song, and you can see that also lowers the pitch of that dashed line there, the bird is correcting the apparent error. His song sounds less bad to him when it's played through the headphones. Then at day 4, after 14 days of this, headphones stay on, but the pitch shift goes back to 0. And you can see that there's the beginning of this return to the baseline pitch. And then we leave the birds alone for weeks and weeks. And then when we come back, and this is actually sort of one of my favorite aspects of this experiment, their pitch has always gone back to where it was on day 0, or almost exactly gone back to where it was on day 0. So this is, important. This is, well, to us, this is important. It proves two things, I think. One is that this process of error, the way that birds are actually maintaining their songs in adulthood, is by constantly listening to themselves and fixing their mistakes. And also, and sort of necessarily, that means that they have this really, really long-lived target that they maintain, at least over 67 days of, me or whoever messing around with the way they hear, that enables them to have this stability. Oh, and this is a little note I write to myself. This is just to remind myself to tell you that this works in both directions. So here we increase the pitch of the feedback or drop the pitch of a song, works just as well the other way too. Okay, so that first experiment, as I said, sort of established that adult songbirds, who've already learned how to sing, are constantly performing this process of error correction, and in so doing, maintaining their song. But there was a lot of really interesting stuff about these data that we still didn't understand yet. So what I'm showing on the right here, this is collected data from that first study I just told you. So I'm changing the format of that I'm showing you the data here in a little bit. So first of all, I'm basically combining upshifted and downshifted experiments so that the Y position, the height of the solid blue line, that's averaged across upshifts and downshifts, and it just means, if it's positive, it just means in the right direction, that the bird is compensating for the pitch shift. Sometimes that's an increase, sometimes that's a decrease, but here I'm combining them so that we can look at them together. And the dashed blue line is just a reminder that this was in response to a 1 semitone pitch shift. So this is really interesting here. So we've replaced the bird's auditory feedback with this auditory feedback that's telling them that they're off by 1 semitone. But really consistently, the birds are only correcting their voices by about 0.4 semitones. And one reason this is interesting is, so I mentioned earlier that I did my PhD doing psychophysics, doing these behavioral studies of brain function. So there I was looking at something totally different. I was looking at how humans plan arm movements. And we had this fancy virtual reality setup where we could use computers to lie to people about the visual position of their hand, just like we're lying to the birds about how their voices sound here. And people will learn if you trick somebody consistently that their hand is to the left or to the right of where it actually is. People will change the way they reach, just like songbirds will change the way they sing when you lie to them about how their song sounds. But humans only change how much they move their hand by about 40%. So this finding where if you just replace a sensory modality and then measure how much the brain corrects for it, it's never 100%, it's never 0%. It's always some number in the middle. So how does the brain figure out what that number in the middle is? Because if we understood that, we would understand how the brain is dealing with this problem of integrating all of these different sources of sensory information along with everything that the brain has learned through, millions of iterations of practice. Okay, so this is the part where I'm compressing most of a decade's worth of work by a ton of people into this list of papers that I'm not really going to talk about in any detail. We did a bunch of experiments to do this, and I'm now going to sort of give you a greatest hits highlight of this. But again, remember that our goal is that we want to just use these behavioral manipulations to try to understand the math that's going on in the songbird's brain when the songbird is trying to decide effectively how to use the sensory information to correct behavior. So I'm going to give you a couple of greatest hits now. So this one really knocked our socks off. Okay, so the blue data the data that's plotted in blue, you've already seen. And I already told you about, that means that if you shift the pitch by 1 semitone, birds will correct for about 0.4 semitones. What's being shown here is a bunch of other pitch shift sizes. So we did something really simple. We just varied the amount of error, the size of the pitch error rather, that we were introducing via the headphones. We saw something really weird. Okay, so first look at blue, that's for 1 semitone. Then we're like, okay, let's try a bigger error. So we used a 1.5 semitone error that I'm showing in green. And learning got slower. So you see that the green line is increasing a lot slower than the blue line, but also it's eventually correcting a much smaller fraction of the total error. In fact, the green is sort of converging on the blue line at about 0.4 semitones, but 0.4 out of 0.4 divided by 1.5 is a lot smaller than 1 divided by 1.5. So proportionally, it's smaller. And then when we did an even bigger error, up to a 3 semitone pitch shift, the bird didn't change his voice at all. That's what's shown in cyan. And then, surprisingly, when we tried a really small pitch shift, which is what's shown in 0.5, the learning is way faster. So there's this inverse relationship between the size of the sensory error that we're introducing and sort of how much and how fast the bird is learning. Okay, here's the results of another paper, where here what we're doing, okay, so on the left it was this experiment where we're taking the same birds and doing different pitch shift sizes. Here's an experiment where we're taking different birds, young birds and older birds, and giving them the same pitch shift error, the same 1 semitone pitch shift error that I showed you originally, and that's what's shown in blue here. These are adult songbirds, and we sort of already knew that their learning curve was going to look about the same here. What's shown in gold there is when we took young songbirds who had just learned their songs, and it turns out they learn about twice as much and they learn about twice as fast. So essentially, and this is what I'm about to say includes some of those papers in that list that I'm not going to have time to talk to you about, is that we collected a bunch of these observations that made us realize that this model that I showed you in the middle here can't be right, or at least it can't be the whole story. Okay, so let's look. For example, look at the plot on the left. What that shows you is, or the data on the left, actually, no, sorry. Let me start with the model in the middle. According to the model in the middle, the bigger the mismatch between the auditory feedback and the auditory target, the bigger the error signal, the bigger the change in the motor system. What the data on the left show is that exactly the opposite of that is true. The bigger the pitch error, the less learning we see. Okay, so the thing in the middle can't be right for that reason. Now think about the model can't be right for that reason. Now think about the data on the right. That's exactly the same difference between the auditory target and the auditory feedback. It ought to be the same error size. It is the same error size. It's always a 1 semitone error. But we're seeing very different size changes in the motor system, in the behavior rather, between young adult animals and adult animals. So again, the thing in the middle can't be right. So what's happening here? I'm going to do a very, very brief summary of this paper because I want to then move on to some other stuff. So here's what we figured, and sort of as computational neuroscientists, which is to say, as neuroscientists who use math to understand the brain, we had to sort of take all of these, sort of apparently conflicting observations and think about what kind of mathematical principles might govern all of them. And I'll tell you about what we came up with now. So so first we want to understand why is it that smaller pitch shifts yield greater learning and vice versa. So the intuition came from thinking about the variability in a songbird's vocal behavior. So here on the left, I'm showing you another one of these spectrograms. This, of course, is a spectrogram of just one single syllable, one discrete vocalization from a songbird. And the arrow on the spectrogram shows, you see this sort of bright band of acoustic power there. That shows the pitch, or the fundamental frequency, we would call it, of the syllable. Maybe that's, I don't know, a high A flat or whatever. Then on the right, I'm showing this histogram, this distribution of all of the pitches that that syllable can have. So you can see the bird doesn't sing at exactly the same time every way, and there's these little variations in pitch. Okay, so this is data just from one example syllable from our data set. Now, don't worry about the units necessarily. I'm going to replace that with this sort of generic blue curve, and just trust me when I say that the variability of this blue curve is about what we see, typically in our data. So what you've got here is, you can see that, when I say mean subtracted, that's like the average. So, most of the pitch variations are sung near the average, and then it falls off as you get farther away. Okay, so remember, the smaller pitch shifts that we saw earlier were the ones the experiments in which we saw the most learning, biggest change in the bird's behavior, were from these very small pitch shifts, which I showed in red. And so what I'm showing you here, the red curve is just the blue curve shifted to the right a little bit. So what the red curve reflects is the distribution of sounds, all sort of the range of pitches that a bird is going to hear through his headphones. And I've shaded in that middle area there to show that there's a lot of overlap between the blue distribution, which is the way the bird has been hearing himself sing the last million times he sang a song, and the red distribution, which is what he's hearing all of a sudden now that we're shifting the pitch. And they overlap a lot. So that means that most of the time, what the bird hears himself sing could have come from his baseline distribution, let's say. On the other hand, as we start doing bigger and bigger pitch shifts, so I already showed you the data, which is that there's less and less learning, but what you can see is an interesting correlate is there's less and less overlap between these probability distributions. And interestingly, remember a 3 semitone pitch shift, that's the point at which learning disappears. So it turns out that you can do a really good job of predicting how much an adult songbird is going to learn by looking at this particular mathematical relationship between the distribution of how he usually sings and the distribution of how he's going to hear himself sing once we start manipulating the feedback. The great part, in addition to that, is that that exact same mathematical model also explains this age-based difference in learning. So on the right here, so just to remind you, remember young songbirds, when faced with the same absolute value of a pitch shift, young animals will learn about twice as fast as older animals. The reason that this, this sort of overlap of the two distributions principle explains that is shown on the right here. So, again, sorry, let's just look at the middle one. So there you've got a pretty large-ish pitch shift, let's say, and you've got some amount of overlap between those two. However, contrast that to what I'm showing for young adult on the bottom, and you have to know this, young adults are about twice as variable as adults here. Young birds make a lot of mistakes when they sing. So what that means is that the width of the distribution of pitches that they're used to singing is way, way higher. And our mathematical model predicts that that increase in variability makes them better at learning because of this overlap, this principle in the overlap of these distributions. Young birds, in other words, are really all over the place anyway. So they're used to hearing themselves sing at this really wide range of pitches, which basically means that according to this particular mathematical model we're proposing, they ought to, show the pattern of data that's shown on the left here. Okay, then I'm not going to explain all this, but I am going to say, I know there's at least one mathematician here, just to say a tiny bit about the formal models that we've been developing. Essentially, and you might have been able, those aficionados may have been able to tell this from my last few slides, we're looking at optimal Bayesian inference as a template for understanding these forms of sensory motor learning. So the idea is basically that we're treating both the bird's vocal motor production and the different schedules of auditory feedback as a prior and a sensory and a likelihood distribution, and then we're iteratively updating a Bayesian model. But you can look at the folks who are interested can check out the papers. Okay, so I've just wrapped up telling you about some studies of learning where the only thing we're doing is we're manipulating how a bird hears himself, and then we're looking at the change in behavior. So what we've been doing has been a way of studying, the mathematics of the brain, where we're not actually directly looking at any brain cells, except through, as they're working together through the behavior of the animal. So for the next part of the talk, I'm going to talk about, I'll tell you about other work in the lab where we are directly investigating the activity of single brain cells and also the muscles that the animals are using to perform the tasks, to try to understand how circuits in the brain are carrying out these kinds of computations that we see playing out in these learning experiments, like the ones that I just finished telling you about. Okay, let's hope. Great. Okay, so in addition to having a really interesting learning behavior, the other reason why songbirds are great to study for us as neurobiologists is that the brain networks that underlie the learning and performance of song in songbirds are really it's really understood where they are and somewhat understood what they do. And they are accessible to study in a way, and then they also turn out to be very, very similar to the parallel brain areas in humans that drive, not only vocal production and vocal learning, but also all sorts of other kinds of motor tasks. So what I'm showing you, this is sort of a cartoon of like a cross section through a bird's brain. In this picture, the bird's beak would be facing to the right, back of its head would be to the left. And these colorful and black ovals I've put on here, these are different parts of the brain. And I'm not going to talk about most of these in any detail at all, except to point out the following, which is that if you look at these areas I'm highlighting in blue and red, there's this anatomical separation in the songbird's brain between the parts of the brain that a bird needs to produce its song and the parts of the brain that the bird needs to learn its song. And those are the latter are the ones that are shown in red. And this is really important. If any of the, let's start with the song production areas. Like if either of those two brain areas that I'm highlighting in blue, for example, if those are damaged at any point in the bird's life, the bird will never sing again. On the other hand, if the areas that are shown in red are damaged at any point in the bird's life, he'll keep singing almost exactly like he sang before, but he can't learn anything new. He can't learn to first sing his song if the areas shown in red are damaged, when a bird is very young. And in an adult animal who's already learned how to sing, if those areas are damaged, birds can't change their pitch in the way that I just showed you with the headphones. So there's this really nice, for those of us who care about, how the brain executes movement and learns new ones in any system, not just those of us who care about songbirds. Songbirds have a bunch of different advantages. So, okay, so I am going to tell you about one particular part of the songbird brain, which is this clump of brain cells called RA. And what I'm showing here is, you see all these arrows that are connecting these clumps of, these parts of the brain. So one of the things that makes neurons, that makes brain cells special compared to other types of cells, is that they send these very long wires to each other that let them communicate electrical signals to each other across these little gaps we call synapses. And what I'm showing here, this is sort of shorthand notation, or in the diagram, I guess, for how neuroscientists draw these circuits. So you see that there's a big old blue thing called RA with a couple of arrows pointing down to those black ovals below. What that means is that there are thousands and thousands of individual neurons in RA, and those neurons are sending these output wires to thousands of neurons in these other parts of the brain that are sort of farther down. And what makes those farther down brain areas special is that those are the neurons that directly activate the vocal muscles rather. So the syrinx is the songbird version of the larynx. It's the vocal organ. And the respiratory apparatus is just what it sounds like. It's the circuits controlling the lungs. So area RA is really, the reason a bird needs RA to sing is that the neurons in RA directly activate the neurons down below it there that contract the muscles that produce the behavior, that produce the singing. And one of the major goals of neuroscience, not just songbird neuroscience, is to understand how those kinds of neurons that are controlling a behavior, how do they control it? What are the patterns of electrical activity that neurons need to fire to produce, a successful version of the behavior? So the way we investigate this in my group is that we manufacture these very, very small microelectrode arrays, which are we can then insert into these particular brain areas to record the electrical activity of one neuron, one brain cell at a time. And I'm going to give you sort of a flavor for what that looks and sounds like. So what I'm showing in the bottom right here, let me explain this. So this is another another graph. On the x-axis is time again. So that bar, 250 milliseconds is 1/4 of a second. And what you're looking at, what those black traces are the voltage signature generated by one brain cell. So there's millions and millions of brain cells in the songbird brain. We're recording from just one of them. And the way, for generations, the way that neuroscientists study these, when we're first recording them, is we just take that voltage signal, and every one of those upward deflections is this particular neuron firing what we call an action potential, an electrical signal. And the time-honored tradition is that to understand or to try to understand what a neuron is doing, you take that electrical signal and you turn it into a sound wave. So I'm basically going to let you play the sound of this neuron firing. And what you're going to hear is pop, 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 pop. Every time that neuron goes pop, it's firing an action potential. So here's what one of these neurons sounds like in a bird that is not singing. This is a bird that's just sitting quietly. Okay, give me some thumbs up if you could hear that. It should have been like ba-da-da, ba-da-da, ba-da-da. Okay, so that's good. That means we're doing a good job recording this neuron, but what we really want to know is how does this neuron control song once the bird starts singing? And this is the most complicated AV part of this talk. We'll see if this works. But let me explain what you're going to see, what you're seeing here. So the bottom trace, that's the voltage recording from this neuron. So you can see, every one of those vertical deflections is the neuron firing an action potential. And then above that is the spectrogram of the bird singing. So what's going to happen when I push play is that the blue bar is going to sweep across to the right, and it's going to go through a few more screenfuls of this. But what you're going to be able to hear is the bird singing. This sounds like the bird song I played you a minute ago. And you're going to be able to hear the neuron firing at exactly the same time. And I hope you'll be able to hear a certain correspondence between what the neuron is doing and what the song is doing. Okay. So I hope there were two things that were apparent there, or at least two things that were apparent there. One of them is that the birds repeat themselves. So the bird, just like people, the bird was singing sort of the same phrase of syllables over and over. You can probably hear that. The other thing I hope you could hear is that the neuron is repeating itself. So in the pop, pop, the sequence of pop, 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 you should have, I hope you were able to hear some repeated patterns. And so what I'm doing here is I'm just showing you what those neural patterns look like. So on top here, this is a spectrogram of, this is this 5-syllable motif that this bird was singing over and over. And then below the spectrogram, I'm showing the electrical recording of that one single neuron on 5 different iterations when the bird sang that same motif. And what I want you to look at is a couple, so now let's look at those 5 horizontal black traces with the little voltage tick marks. So I want to point 2 things out here. One of them is that there's a lot of structure across these different renditions here. So you can see that in every one of these 5 examples, the neuron is firing some action potentials at roughly the same time. But then you'll also notice that if you look within a certain burst or a certain little cluster of those, you'll see that the number of action potentials and the timing of the action potentials actually varies substantially from iteration to iteration. And again, this is about to be about a bunch of work that I'm not going to go into any detail about. Another big part of the lab is trying to figure out how those variations in neural activity that you can see there, that you can see in the middle there, how are those controlling the variations in pitch or loudness or any one of these other acoustic properties that songbirds are controlling and that we've already shown songbirds are able to modify through experience. So again, this is yet another, more for the mathematicians in here, but again, we're using another set of Bayesian techniques. I want to tell you just very, very briefly about one mathematical technique that we've developed, and hopefully this is going to be published soon, which is that, so you remember, for everybody, you remember in the recording I just played you, there were these really, diverse and interesting patterns of action potentials. This is what I'm showing in the top left here where it says spikes. And so what you have here is you have something that has structure in it. The spikes are happening roughly at the same time when the bird sings, but also have a lot of variation in it, which is, there's differences in the number of spikes or timing of the spikes across each iteration. So this is something that we've done, my group has done in collaboration with Ilya Nememan, who's in both the biology and the physics department, is we're creating mathematical tools to basically identify the important patterns in any complex data set. So what do I mean by important patterns? What I mean are the patterns, the patterns in the data that most predict the outcome of the output of the system. So in our case, it would be patterns in the brain activity data that most predict the behavior. But you could also imagine, this was shown on the left here, that you have exactly the same problem in other fields of biology. So for example, you want to predict how a genetic sequence predicts the function of some protein or whatever, some system in the body. So sort of the excitement of this technique again, I hope I'm doing a good job of presenting this, is that we are interested in songbirds. Songbirds are great, but the kinds of tools and approaches that we're trying to develop really go a lot beyond just understanding vocal control in songbirds. I think I don't have time to go into any of the details here, but this is I think a nice example of that. Okay, so Ron mentioned very briefly the consortium that I'm the director of. I just wanted to spend a couple of minutes talking about this. Actually, and this leads nicely from, or rather follows nicely from what I just said, which is that songbirds are great, but we want to understand, how the brain controls behavior in a much more general and sort of a much more general way. Okay, so the Simons-Emory International Consortium is a very unusual project. Very grateful to Simons Foundation for providing funding for this. So what we are is we are a network of 8 labs in the US, Canada, and Portugal. And we all work on really different animals and really different behaviors. And we are, as Ron mentioned, we've sort of come together to create, a common tool set, but also a common mathematical language for thinking about how these very different behaviors, which are being shown here, in really different species are controlled. And, I know that there are very few, if any, neuroscientists in the department, sorry, in the audience here, but as academics, I imagine it will not surprise any of you to hear that people who work on one part of neuroscience don't really communicate that well with people working in another part of neuroscience. And sometimes it happens that results in one subfield are not directly comparable to, anyway, you get the idea. So, and frankly, one of the reasons that happens so badly in neuroscience, I think, is that, let's say I study songbirds, I don't go to meetings with primatologists. So, you can have the same, good or even not so good ideas propagating at the same time through fields of people, through subfields where people don't talk to each other at all. And, what we did here, this is very different from the kinds of sort of large, big science consortiums that are kind of popular in neuroscience right now. Those are generally, focused on, 10 labs around the world doing exactly the same experiment in slightly different ways. Our idea here is that although the mathematical tools and some of the hardware tools that we've developed are in common, everyone is looking at a totally different aspect of brain function, but within this common mathematical framework, so that we can then talk about, how is it that, walking in a mouse is similar or different to singing in a bird, from the perspective of the brain. Oh, okay, sorry, I meant to do this one as well. So I just also wanted to acknowledge, in addition to these, I've been focusing mostly on mathematical tools. We also have been developing different hardware, new kinds of electrodes, basically, ways of, in a minimally invasive way, recording the activity of muscles in any of these species that we're working on. And that support came from, first from the McKnight Foundation. Okay, Gray, how am I doing on time? I have an option to jump out here, or I could...
Oh, you've got plenty of time.
Okay. Okay, great. All right. So I'll talk about one more. This is good. This will be good. This is in a little bit more detail than I was presenting before. Okay. So now I want to talk a little bit more about another way that we use the Songbird brain to investigate the different brain mechanisms of learning. So this is what I'm showing here. I showed you a slightly different version of this figure. This is a sort of a cartoon schematic of the Songbird brain. I'm sorry, I changed the color scheme here, but the two areas that I've highlighted in orange here, HVC and RA, which we already talked about, this is what we refer to, just as a reminder, as the motor pathway. So this is the part of the Songbird brain that is necessary for producing song. And then the parts that I've highlighted in green here are this learning pathway that's necessary for learning. Another thing I mentioned earlier in the talk that I'm now going to come back to is that there's a very well-studied correspondence between different parts of the songbird brain and different parts of the primate human brain. And it turns out that they have, roughly speaking, the same types of brain cells that seem to be performing analogous functions in terms of learning and the control of complex behavior. So that's actually made the songbird also really attractive as a way of trying to understand particular brain systems, including those that are affected by different kinds of neurological disease. So I want to point out, okay, so I was talking, in the previous section, I was talking about area RA, so that's where I played that neuron whose recording I played you was from RA. Now we're focusing on an area called area X, and area X is in the songbird basal ganglia. And one of the big questions, so we know from these behavioral experiments, like with the headphones, that birds must be processing, they must be using the sound feedback to change their behavior because, if you play them sound that makes it sound like they're singing too high, they'll lower the pitch of their song and vice versa. So something in the songbird's brain must be saying, hey, I just made a mistake and I need to change something. And what happens, surprisingly, and we also knew from other studies, like these ones where if people would experimentally damage one of these parts of the brain, we knew that these parts of the brain were important for learning or producing song. But then when people did those neural recordings, like the kind I just showed you an example of, people would do stuff like they would record from a neuron in any part of the brain that I've shown here, and then they would play some weird sound that sounded like the bird making a mistake, and the neurons didn't care at all. So this really confounded the field. They were like, people had gone to all of this effort to identify all of these areas that are critically necessary for the process of song learning. But then when people actually started to record the activity of most of those areas, it turns out that those neurons did not appear to care. They didn't change their firing at all when the bird heard a mistake. So then this question becomes then, how did these signals about error, so we were talking about these error signals, say the difference between what a bird hears himself sing and what the target, what he wants to hear himself sing, how do these errors get into the song system? How do they reach these parts of the brain here? So I mentioned that this part of the brain called area X is part of a larger structure called the basal ganglia. And one thing that is very distinctive about the basal ganglia, and this is of course it's true in humans, as I'm about to get to, is that parts of the basal ganglia get lots and lots of input from the neurotransmitter dopamine. So what I mean by that, so first, what do I mean by a neurotransmitter? So I mentioned that what makes brain cells special is that they can communicate with each other across these little gaps called synapses. Neurotransmitters are the chemicals that cross that gap to contain the messages. And there's lots of different neurotransmitters. They all have different functions in the brain. But this dopamine input to the basal ganglia is very important for lots of forms of learning and behavior. It's also, as many of you may know, this is the population of neurons that dies off in Parkinson's disease. So a lot of the symptoms of Parkinson's disease are attributable to a loss of dopamine neurons. But interestingly, these dopamine neurons in songbirds that project really powerfully, there's lots of these projections to area X, this part of the basal ganglia that's involved in song learning, they've never been considered part of the song system before. Nobody actually knew whether they had anything to do with learning. So one of the other projects that we've been doing has been exploring this hypothesis that basically this group of dopamine neurons, this tiny little group of dopamine neurons way deep in the brain, that those are the neurons that are providing the error signal to this learning network. It's basically that the dopamine neurons are the ones who are saying something wrong happened, you need to change your vocal production. So we use lots of different techniques for investigating dopamine neurons. So what I'm showing here on the left, the sort of tan image in the background. So that is a very thin section through a songbird's brain. The intensity of the brown section there shows how much it's what we're actually staining, where we're coloring something brown. The thing that we're coloring brown, it's not dopamine, it's the enzyme that makes dopamine. But basically, you can just think where there's brown, there's dopamine. And so, can you guys see? Y'all can see my cursor here, I hope. Great, cool. Okay, so down here, this is this tiny little part in the brainstem, in the midbrain. This is where the dopamine cell bodies are. And then remember, each neuron will send a long wire to its targets. These wires ascend. You can actually, looks a little like hair here. That's these very fine axons, or very fine wires projecting up. And then this area up here that's stained really dark brown, that is the songbird basal ganglia. And what you can see on the right here, I won't explain this in too much detail, is that we can use different kinds of chemical and genetic tools to isolate these neurons, to visualize them, and to sometimes manipulate them. Okay, so we have this hypothesis then that these particular neurons, and not just these dopamine neurons, so this is actually another very important thing. So dopamine kind of goes everywhere in the basal ganglia. It goes almost everywhere in the brain, and it definitely goes everywhere in the basal ganglia. And dopamine does lots and lots of different things. This is one of the things that makes Parkinson's a challenging disease to treat, frankly. So what we wanted to do is we wanted to test not a very specific hypothesis, not the hypothesis that dopamine is important, because dopamine is always important. We wanted to test the hypothesis that the particular set of dopamine neurons that provides input that provides input to area X, this part of the learning circuit, that those are the particular neurons that are conveying the error signal. So what we did is we used this pharmacological approach where we could use it to specifically kill a tiny subset of the dopamine neurons. We estimate that we're killing about 1% to 2% of the dopamine neurons that otherwise would have provided dopamine to this part of the basal ganglia. And as far as we can tell, and we checked in a bunch of different ways, we're not hurting any other part of the brain. So the intention, at least, is very specific lesion just to kill a few dopamine neurons to test the hypothesis that that's the signal that birds use to learn. So to do this, although we've actually gone back and did this with headphones, we actually used a slightly different learning paradigm here that does not involve headphones, where basically the bird sings and then we can use online computer analysis to detect when a bird is singing a particular pitch. And then if the bird is singing above or below some particular threshold pitch, we can then play a blast of white noise. Birds don't like that, and they will learn how to change the pitch of their voice to avoid that white noise. So it's different in its technical implementation than the headphones, but it's still a bird using auditory feedback to figure out how to change its voice. I'll skip the details here. This is just showing in the top right, you can see that area X that normally is a darker brown, we've made it into a slightly lighter brown. That's a sign that we've just killed the dopamine cells and nothing else. And then the key result here is what's shown here. So let's look on the left. So the birds are undergoing 3 days of this auditory training where they learn how to change the pitch of their song. Black is the bird's behavioral data, so how much the birds learned before we killed some of the dopamine neurons in this part of their brain, and red shows after. And you can see that by removing, and I should mention, we measured how many of the dopamine cells we were able to kill, and our best estimate is that we killed about half of them. And it also turns out that learning still exists after the dopamine lesion, but it's only about half as strong. And then the plot on the right just shows that in a control sham experiment where we just go back and retest the birds without having injured the dopamine cells, they stay just as good. Okay. So then just to wrap up, what I hope I've convinced you of, or at least given you a flavor for, is that songbirds and sort of increasingly now these other species that were part of this consortium provide a really great way of looking at these very complicated relationships. So the relationship between patterns activity in brain cells and muscles, the behavior that they produce, and then the way that sensory feedback from behavior is used to sort of rewire the brain during sensory motor learning. I also want to just close by saying that this has been the work of lots and lots and lots of people, and that one of the things that's been great about being at Emory has been starting new collaborations here, and also using here as a springboard to set up collaborations elsewhere as well. So I will stop there.
Thank you so much, Sam. And now for people who have questions or comments, if you will raise your Zoom hand. Vernon has his hand up.
Yes, thank you so much. This is amazing. It just happens that on March 11th, I downloaded a BirdNet with the recommendation of a friend, and I see I have 160 bird observation recordings since then. So I've been just ironically following these. My question is the relation of purposive bird singing to what you're doing. In other words, do they vary? The tufted titmouse will communicate with another titmouse. Are they varying their pitch purposely to give a particular identification to the other titmouse is coming? Also, Carolina wrens do an interesting sequence.
Yeah, they're so small but so loud. So let's start with the purposiveness. So one thing that's very, people, including me, use songbirds to study this is what we call social context dependence. So a songbird, and the song, I mentioned territoriality. Most of most of the time when people are studying birdsong, they're studying in the context of courtship. So it's more sort of as a mate selective performance. But here's an interesting fact, which is that when male songbirds are singing to female songbirds, they have very, very narrow variability. They actually shrink their variability to be as precise and as repeatable as possible. When they're singing by themselves, the variability doubles. And there are lots of theories about this. One theory is that, when they're singing by themselves, it's practice, and they want to really explore all the different possible variations. Who knows, maybe they'll find something better, but they're not being evaluated by a female on it. So now whether you think that that's purposeful, I don't know. It's consistent with a purposeful story. I don't know if it's really purposeful. The other thing, though, is sort of what is being communicated. So for individual identity, I know less about that. There's been some work on like what aspects of song do females prefer, and that's consistent with two things. One of them is having low variability, which makes sense. Like if you give a female bird a choice between listening to a low variability song and a high variability song, they always pick the low variability one. And then in cardinals have their so-called sexy syllables. They're these really, really acoustically complicated syllables. Female cardinals prefer those too. But yeah, as far as, for territoriality, and I'm sort of speaking outside of my expertise here, I don't know whether it's, hey, get out of here, or like, hey, get out of here and my name is Sam. You know what I mean? Like, I don't know whether the signal whether it's important to name that it's specifying an individual or just telling somebody to beat it.
Well, would it be fair to say that purposive study of birds has helped to kind of lead to the kind of precise cognitive studies that you're doing?
Yeah, I'd say so.
Lies
Sure.
in the background earlier.
Yeah, I think so. Yeah.
Thank you. So good.
Okay, Rod Hunter has his hand up.
Thanks for a wonderfully fascinating talk, just so interesting and clear. 15 years ago, when I retired, I returned to one of my early childhood loves, which was classical piano studies.
Okay.
And so much of what you say rings true to things I have learned about learning in the 15 years of piano studies that I've taken in subsequent years. And just a couple of points quickly and then a question. One is that you learn when you break things down to very specific units. You don't want to take a big chunk. Much of what I do is memorization. You don't want to do a big chunk of material. You can get a gestalt for it and then it'll fail. You have to break it down into little pieces and learn and think about each little piece until it becomes automatic, and then they all go together. If you make a mistake, you don't do a running start ahead of the mistake to try to clear the hurdle. Yeah. You start right on the mistake and try to correct it. But one of the most fascinating things to me in the early days of my recovering piano experience was when the teacher said to me, "You are memorizing your anxiety."
Okay.
That was one of the most insightful things I think I've ever learned about it. And I've since learned that effective learning of the piano does require keeping anxiety very, very low. Sure. And that means you do things deliberately, slowly, and try to get them correct. And you don't worry about speeding up. You don't worry about performing. You don't worry about anything. You just practice it. So the question here is there anything, it's tempting to want to hear your studies as pointing to some aspects of human learning. And I'm wondering if there's anything in your studies that relates to this principle of low anxiety, of
that's interesting.
Birds. Calm down. And
well, I mean, so yes, for, relating to human studies, so I talked about, difference in learning dynamics between younger and older animals. And there's a whole, in the speech, the speech literature, and that's, both basic science research on speech and also some stuff about, speech rehabilitation. Yeah, there's a bunch of parallels between birds and humans there. Not so much about anxiety. It's hard to, I mean, we would need some sort of readout of bird stress levels, which, I can imagine what they might be, but we haven't done that yet. But yeah, it's a good idea.
Thank you.
Yeah.
Okay, Thrivi has his hand up.
What I wanted to know is, you showed that VTA to basal ganglia projection,
Mhm.
dopamine project to from VTA to basal ganglia. But
Yes.
how does that project to area X in the cortex?
Area X is inside the basal, is part of the basal ganglia. But there are other dopamine neurons that project from VTA into cortex. It's just a separate population of cells.
So when you destroy those basal ganglia neuron, you assume that those neurons are also destroyed?
Only, no, I didn't explain this in detail. We use a particular chemical agent that only attacks dopamine neurons. It doesn't attack any other kind of neuron. And we do, lots of validation studies to show that, when let's say this is the sphere of tissue that we're injecting the drug into, it only kills the very small number of dopamine fibers in that area. It doesn't kill it. It doesn't seem to hurt anything else.
What about the reward pathway?
Well, so the idea is that this is the classical reward pathway. And, the idea is that, just like dopamine is thought to be involved in processing, all kinds of, hedonic rewards and, things associated with drug addiction, that it's basically, the idea is that it's basically the same neurochemical process that underlies the reinforcement of, desired vocal outcomes. And there's lots of precedent for thinking that's true in some human studies also.
I have a couple of more questions if you bear with me. Is male and female sing the same way?
Right, great question. So in the particular species that my group studies, only the males sing. There are a number of species where both males and females sing, and it's very interesting. So these brain areas, so they're under in the finches that we have where only the males sing. During development, these areas undergo massive increases in size in males due to hormonal fluctuations. But in species where both males and females sing, it's the same set of brain areas in both males and females. Yeah, actually, some species will duet. It's really cool. So they're both song learners, and there's actually a dance that goes along with it.
So what is the difference between basal ganglia projections between male and female?
Great question. It's hard to say because, okay, so in the non-song area, so remember the basal ganglia does lots of things, not just song. In the non-song areas, everything, as far as I know, is identical between males and females. But then in the species where females don't sing, the analogous area is simply not present in the female brain, unless it's a species where the females do sing. So we can't. It seems, and the thought is that this particular sort of module of brain areas evolved as a reduplication. So a reduplication of a brain wiring motif that's already, there are already many of these in the brain. So, it's a loop. I mean, it sounds like you know something about this. The loop between the cortex, basal ganglia, thalamus, and cortex, that's reiterated. There's lots of different brain systems that work that way. The idea is that in whatever species that evolved song learning, the song system, these parts of the brain that I've been talking about, evolved as just one more reduplication of this sort of motif of cortex, basal ganglia, thalamus, cortex networks.
So that means in Parkinson's disease, speech abnormality may not be attributable to basal ganglia.
It's possible. I mean, so there are studies of people being on and off L-DOPA. So I mean, obviously, like Parkinson's is multifactorial, and it's also a slow progressing disease. But there are differences actually, and this is for both people with Parkinson's disease and people who don't have Parkinson's disease. You can give them Parkinson's medication effectively, which will modulate, global, dopamine levels, and there are speech consequences of that. Of course, it could be that some of the vocal consequences of Parkinson's disease don't have anything to do with acute dopamine loss and might be some secondary effect or something else.
Thank you very much.
Okay.
Kerry has his hand up.
Thank you very much for the wonderful, enlightening
talk. Opens up a lot of possibilities for even us non-scientists who are avid bird listeners and have been for some years. And one of my question has to do with something that we've heard over a long period of time. That is two sounds that recur from the different birds. I believe they, maybe a wren possibly, but one of the sounds that we've heard is, just repeat the sound, is receipt, receipt, receipt. Another one is reacher, reacher, reacher.
Okay.
And my question has to do with how they have arrived. Now, this has been going on for some.
Sorry, so you're talking about like what the bird song sounds to you as
That's right.
phonetic
Translated
English. Okay.
into
Got it.
what we can identify, though we don't really ascribe it to
Sure.
an English identification, but it's the sounds that we recall. And the question has to do, and you have answered this in various points of your talk, and that is how they arrive at those particular sounds, those particular notes.
Right.
Over the years, how did they come? Where did they hear that, and how do they repeat the sounds? And I realize you have, in a lot of ways, laid this out, but
Well, I mean, me and lots of other people. I mean, so I'll tell you about some classic work on this. That this is not my lab stuff. But so here's an example. So okay, let's just, I'm just sticking to the species that I actually know about. So in the species I actually know the most about, only the male birds sing. So basically what that means is that the particular song syllables are actually culturally transmitted, which is that they're transmitted from, in this case, father to son to father to son, with small variations each time. What that means is that there are there's different bird colonies will have, basically, it's inherited behaviorally, not genetically, the same way that languages are inherited behaviorally. But then, okay, so this is the cool experiment I want to tell you about. So, so the reason why the bird you heard was saying reacher, reacher, reacher is because its dad was singing reacher, 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 and that's what it learned. I know that's not what your exact question was, but the experiment people did is they took a whole bunch, they did the desert island experiment. So they took a bunch of chicks, reared them without fathers, which is to say without singing. They produced this crazy song, that was already known, but then they basically kept them on the desert island for generation to generation. And what eventually happened was that over, I forget how many generations, the acoustics went back to reacher, 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 basically. So, and I mean, one of the things that's really neat about birdsong is that it illustrates both, the incredible plasticity, the incredible changeability of vocal behavior, but nevertheless, there's this, really sort of slow timescale genetic background. Oh, here's another example. Maybe this illustrates it even better. Okay, so this is Peter Marlar's work at UC Berkeley in the 60s. The way that it was realized that songbirds had this cultural transmission is that in the Tennessee Valley, just north of San Francisco, there are these different, it's all the same species of sparrows, but they're divided into these different micro-populations by the hills, basically. And this is only made possible because there were new audio recording technology. Marlar just recorded a bunch of them individually and found out that it was a different dialect in this valley as in that valley. And then what he did was, after that, was a bunch of cross-fostering experiments where you could not only could you take a chick from Valley A and put it in Valley B and it would learn Valley B's song, you could actually do it across species. You could take a chick, an egg from species A, and give it a choice between its own species or another species to learn from. And if you do that, it will always learn from the species it has direct social contact with.
Oh.
So they can learn other species' songs, no problem, as long as it's the one that they're in social contact with. But if you give them a choice between two tape recordings, they can never have seen a member of their own species. They always copy the song of their own species.
Oh, wow.
So yeah, I mean, it's a mix. This is sort of really awesome classic work on, genes versus sort of the flexibility of the environment. So my guess is that there is something genetic about reacher, 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 but if you took a bunch of whatever those birds were, isolated them from each other, they would sing something crazy for a few generations, but then they would probably go back to reacher, reacher, reacher.
Thank you very much.
That might have been a longer answer than you were expecting, but yeah.
Don't know.
Okay.
Absolutely.
Larry has his hand up.
Yes, great talk. I have a question about birds like mockingbirds that mimic the sound of other birds. How does that fit into your paradigm of a bird, never changing once it's an adult?
So there, yeah, great. So there, all right. So again, this is a good point that keeps coming up. My lab only studies really two, but really mostly just one species of songbirds, which is unusual, or at least not representative of all birds. So the birds I study are what are called closed learners. And what that means is that they have this period early in their life when they do most or all of their vocal learning, and then the capacity for vocal learning closes, basically. And birds are, they can still do the headphones learning, but they can't learn a brand new song like they can when they're young. And one of the reasons we study those birds is that's most similar to human speech. Like, you can learn a language in adulthood, but it's much easier to do it in this early, we call it a critical period. On the other hand, there's lots of birds, none of whom my lab happens to study, that are what are called open learners, and that they can learn new things through this process of vocal copying throughout their lives. And so, any bird that imitates a car alarm is an open learner, unless it was raised by cars, which probably didn't happen. But then, for example, there are seasonal open learners. So this is blanking on the particular species, but many species of songbirds will learn new songs in the spring, in time for courtship and mating season. And there's lots of interesting changes in songbirds' brains that go along with that seasonal addition. So it's a little different from a mockingbird, but for seasonal learners, there's a bunch of neurogenesis, which is to say a bunch of new brain cells are born and are incorporated into those areas that I showed you before. So I guess the short answer is that yes, there's a lot of study of sort of adult brain learning and a lot of really interesting brain correlates of that, but people mostly do it in seasonal songbirds. I think mostly just because they're easier to work with in the lab.
Thank you.
Gretchen.
Hi, Sam, and I too want to thank you for a really wonderful talk.
Thanks.
So this is my question. If a bird, say, flies up outside my window, lands on a branch, opens its mouth, starts to sing, is it ever the case that it's singing just for the sheer joy of being alive?
Ask it. I don't know. Sure.
I like to think that, but it sounds doubtful that is the case.
I don't know. Yeah, I mean, this is more speculative than I normally get. I'm not sure birds have hobbies. I believe that some mammals have hobbies. Humans definitely have hobbies, but birds are really all business. I'm not saying birds don't have fun when they're singing, but the particular birds are old. Here's a physiological fact about birds that has nothing to do with neuroscience, which is that basically birds are almost always just on the point of starving to death. They have chosen this way to navigate the world, where they can fly, they can get away from stuff, but all their bones are hollow. They can't carry hardly any fat reserves, and they don't have time to waste. So I think of birds, and again, this is way more anthropomorphizing than I normally let myself do in a seminar. I think birds always have somewhere to be. I think my dog has hobbies. I don't think birds have hobbies.
Well, as Vernon was saying from early on, it's all purposive in practical ways, which means I can see why the goal is to stabilize the song and maintain that through the adult existence. But I guess that means there's just no space for creativity.
No, I totally disagree.
No. Good.
Yeah.
Please,
no, I mean, it's all creativity aimed at passing their genes on and not starving to death. But no, I mean, I would actually argue that the songbird brain is the best biologically understood mechanism for creativity, which is that
say a little more about that.
Well, I mean, it's just that I strike from the record everything I said about all the anthropomorphizing comments. No. I mean, it's, what do I mean? I think that, the reason I got into studying birds is because I'm interested in the relationship between variability and learning, which is sort of, how do you constantly try to come up with new stuff while still having a decent average performance? And, in development, for example, in young songbirds, and I assume young kids, first of all, they don't know what they're doing, but the way they deal with that is by having these really high levels of variability that let them, I think of that as being not that different as from creativity. I mean, they're constantly innovating to see what works better. So I guess what I mean is that, like, I wonder whether it's not that I don't doubt that animals can experience the emotions like joy. It's that I doubt that that's a bird emotion.
I'm glad to
Don't tell my department chair. No telling my department chair that I just said all this stuff.
Thank you.
Okay, Ron.
Yes, let me thank you, add my thanks for that wonderful, interesting, and informative talk. And I have a serious question, but my first is just a little something that happened years ago when I was going down south, and there was stopped by the side of the highway to a rest stop, and there were a bunch of crows on the telephone lines. And when the traffic would increase, I was certain that I heard them call car.
That's funny.
Anyway, my serious question has to do with your many years ago, when I was peripherally associated with neuroscience, but you were talking about the songbirds, the acquisition part and the production part of the song. Actually, these are sort of two related questions. And you had the acquisition part in the anterior portion of the bird's brain, and the production part posterior. Normally in the mammal's brain, we think of the sensory part as being posterior and the motor part being anterior. But this relates to my second question, which was your recording of that RA portion of the brain. It looked to me like they were silent when the bird was making its song, and active when it wasn't. So my question is is that brain portion of those cells that you were recording from, were they not acquiring?
No, they were. You're definitely right that there was a big change in their firing pattern, when the bird was not singing versus not singing. But when the bird's not singing, it's that very metronome, like pop, 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 pop. And then when the bird starts singing, it's producing about the same total number of action potentials of electrical events. But during singing, they get grouped into these bursts that are really close together. So instead of being pop, 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 it's like ba-da, ba-da, ba-da. So it's not the one of the things that's interesting to me about it is that it's not that the overall level of activity really goes up or down that much. It's that this sort of the timing intervals are what change really dramatically.
Thank you.
And Vernon.
Yes, Sam, thanks again. This took a very interesting turn when Gretchen raised the question of creativity.
Yep.
And this reminds me of the context where I, as a teacher, I was trying to help students be creative, because that's one of the things Emory University does. Right? So the question is, do you let students do what they want to do? Okay, that's kind of one form of creativity. Then I read some studies that said, well, if you create just a little box for the students and then raise questions that are essentially asking them to find their way around there or out of that box, then suddenly it really energizes creativity. Now, it's your description of the birds as always having to be doing something to find food. Sounds like the latter, namely that they are in a little box, and you say, oh, no, they're very creative. In other words, they're finding ways to deal with that reduced environment.
Yeah, that makes sense to me. Yeah, no, I mean, they've got, one thing, I mean, this is more stuff I didn't talk about, but, yeah, the Songbird Brain is extremely good at sort of, you can think of it as exploring a space, but it's not a geographical space. It's the space of all possible patterns of brain activity, basically. And, you can, like, you want to, I mean, and maybe the analogy to students is, you want them to be exploring the right neighborhood. It doesn't matter if they're very accurate at first, but like, whether it's a pencil and paper problem or something that you should have like a pottery wheel and a, bowl of gumbo for, you got to get people into the right neighborhood.
Well, I'm wondering if we're talking about two modes of creativity.
Sure. Yeah.
Trying things just through trial and error, just and try to help them have some of that joy that Gretchen is hoping they're having, just to explore and try.
Yeah.
And then the other is then get into this restricted area and say, can you get out of it? Kind of.
I see what you mean. Yeah.
Thanks.
Angelica.
Thank you for a fascinating talk.
Yes.
I confess I'm a little, shall I say, freaked out by the idea of headphones on a bird, but whatever. So here's my question. Since when birds sing, it's a communication to other birds.
Mm-hmm.
So I wonder, when you change the song slightly, or like in the valleys in California where there are different dialects, do they understand a different dialect? And how?
Yeah, so thank you for asking that question. So this is very cool work, not done by me, but very relevant to your question. So yeah, there's a very nice way, happens to be a friend of mine, did this experiment, is you train a female bird to distinguish the song of two males. Basically, female birds already do this all the time, but you teach the female bird that if she hears male A, you jump on this perch, and if she hears male B, you jump on that perch. So it's not that you have to teach the female bird to discriminate, it's that you have to teach the female bird to tell you what she hears. And then you can start manipulating the bird's sounds, basically, and see, at what point can she no longer tell the difference, basically. And it turns out that, so two things are important. One of them is the frequency. So if you shift I forget the exact number, but if you shift the pitch, a few notes, basically, the bird won't recognize it anymore. And also, if you speed it up or slow it down by a certain amount, they'll stop recognizing it anymore. So that's, those are really nice studies. That tells us that those are two things that birds are using. It doesn't tell us sort of the full list of things that birds are using, but it tells us, pitch matters and time matters, basically.
That's so interesting. Thank you very much.
And Ron Gould.
Sam, I was fascinated from the beginning by the headphones.
Okay.
I'm assuming you didn't just go to Best Buy and say, I need a pair of bird headphones.
No, you can't. Well, here's something funny. So the whole problem, I am not a mechanical engineer. The critical thing was the hearing aid industry. So I could not have built a Songbird-sized headphone from scratch, but because zillions of dollars have gone, you can buy a really good earphone, in-ear hearing aid speaker for about $30. So the easy part was the speakers. Everything else, it was the ergonomics that were tricky. Anyway.
How do you get them to stay on the bird?
They have to be very comfortable. They have to be pushing in hard enough to create an acoustic seal, but not hard enough because the birds won't sing if they're not happy. So you can't, I think the first ones may have been a bit too tight. But yeah, you got to find the right level of pressure. And then you got to find exactly the right kind of foam rubber earphones to cut out with a hole punch to make the little pad, stuff like that.
You've overcome some interesting problems.
Tell me about it, yeah.